My name is Angel Rodriguez, and I'm an instructor at the Lester and Sue Smith Breast Center at Baylor College of Medicine, Houston, Texas. And on behalf of Dr. Jenny Chang and researchers at Baylor College of Medicine, I'm delighted to be presenting our results of a study that looked at the relationship between tumorigenic cancer cells, or breast cancer stem cells, and breast cancer treatment. The field of cancer stem cells research is an exciting new field that, although not fully accepted, it's fairly new. More research has been done in the last 5-6 years. It has been getting more attention. Great. So to understand our study, one must first understand the concept of cancer stem cells. This concept has two major components. And these are that, it's not true, that cancer stem cells are self-renewing, meaning that they can make exact copies of themselves, and that they can form tumor on transplantation. These cells are thought to compose approximately 10% of the whole tumor. This theory is certainly not new. I think it's worth mentioning and recognizing a man who introduced the concept of the cells stemming from other cells, German doctor Rudolf Virchow, the father of modern pathology. Exactly 150 years ago, in 1858, he presented a series of lectures, brilliant lectures, at the Pathological Institute here in Berlin. In these lectures, he presented his famous theory, omnis cellula est cellula, where every cell originates from another existing cell like it. And this is basically the cornerstone of the cancer stem cell hypothesis. In 2003, researchers from the University of Michigan presented a landmark study in which they identified this subpopulation of cells that, when transplanted into immunocompromised mice, they were able to generate a tumor when transplanting very few cells. This is a diagram of the breast cancer stem cell hypothesis and the effect of treatment on stem cells. Previously, in a prior study, we had shown that chemotherapy actually increases breast cancer stem cells. In addition, we showed that there was increased tumor formation when tissue samples from chemotherapy-treated patients were transplanted into immunocompromised mice. In this figure, figure A shows how standard therapies can eliminate the bulk of the tumor, and one might see very good regression of the cancer. However, the black circles, the so-called breast cancer stem cells, may remain, and this may lead to recurrences in patients who have been treated with chemotherapy. This may explain why 1/4 to 1/3 of the breast cancer patients have recurrences. In Figure B, treatment that targets the tumor breast cancer stem cells may not even show regression of the tumor, and in Figure C, a combination of both treatments may completely eradicate the tumor. So the question arises, are these cells an important target in cancer treatment? Are we missing these targets with our current treatments, and should we target them? Are there currently any treatments that inhibit these cells? Well, in Baylor College of Medicine, we completed a phase 2 non-randomized open-label study to determine the clinical efficacy of lapatinib. Lapatinib is a small molecule which inhibits an important target in the cancer stem cell self-renewal. Basically the EGFR HER2 enzyme. It has been licensed in the United States in the use in metastatic breast cancer patients, and I believe it's under review here in Europe, maybe with temporary license in Switzerland. So lapatinib was given as a preoperative therapy as a single agent in patients with treatment-naive, locally advanced or metastatic HER2-positive overexpressing breast cancer. They received 6 weeks of lapatinib at 1,500 milligrams orally daily, followed by conventional therapy, which is trastuzumab and docetaxel for another 12 weeks. Within the treatment of the lepanib, we performed biopsies every 2 weeks to look at the effects of just molecular effects of the treatment as well as cancer stem cells. The median size of the tumors in our patients was large, about 10 centimeters, which is the size of a large orange. We had 45 patients. With this treatment of 6 weeks of lepanib, we saw a median decrease in size of 50%. And we achieved a 75% clinical or partial complete response, defined as at least a 50% reduction in the product of bidimensional tumor measurements. A couple of pictures to demonstrate what we saw. On the right is a tumor before the therapy, very large, maybe the size of 6-7 centimeters, and after only 6 weeks of lepanib orally, we saw a marked regression of the tumor, as evidenced on the left. Some clinical pictures of the tumors. On the top, we see a ulcerated, what we call inflammatory breast carcinoma, and on the left, we see resolution of the skin changes, and the same thing on the bottom, large inflammatory breast cancer with also large mass, and on the left, a almost resolution of the tumor itself, but definitely changes of the skin back to normal. So we took biopsies of the first 30 consecutive patients enrolled in the study to analyze the effect of lepanib on cancer stem cells. Through the use of flow cytometric analysis, we analyzed what happened to the effects of these stem cells. And we saw the opposite of what we saw with conventional chemotherapy. As I said previously, we had shown that chemotherapy increases cancer stem cells, whereas with lepanib, we actually saw the opposite. We saw a decrease of cancer stem cells. Another functional assay that we use to analyze these stem cells are mammospheres. And mammospheres are basically clusters of cells that one sees when you grow them in suspension. And this is a functional assay that analyzes the capacity of these cells to self-renew, to make more cells. And we also saw a threefold increase with chemotherapy, but we saw a decrease with lepanib. So now what was the effect after with surgery? What did we see? So the patients that completed their standard treatment with 12 weeks of docetaxel and trastuzumab, we saw these effects, and the treatment achieved an excellent response rate. We saw almost 2/3 of our patients achieved a pathological complete response or a near pathological complete response, defined as minute foci less than 1 centimeters. And these are very exciting and very encouraging news. Let's see, and so what do we get of all of this? Well, this is one of the first studies this is the first study that looks at the effect of treatment on cancer stem cells and show that they decrease with a certain treatment. So first thing, we need to be obviously cautious with the results. I think that number one, more study needs to be done on these cancer stem cells. We saw basically what we can get out of this small subpopulation of cells with tumorigenic potential is intrinsically resistant to conventional chemotherapy. The dual kinase inhibitor lepanib may affect self-renewal of these tumorigenic breast cancer cells and sensitize them to subsequent chemotherapy. And specific inhibitors to pathways that are important in breast cancer stem cell renewal may enhance efficacy of current therapies. Thank you.